Thank you so much again for Thank you for joining this afternoon. Really, I suggest that that you buckle your seatbelts. We're going for quite a ride into the new frontier of treatments for ALS, and really the speakers need no introductions. I'm gonna keep my introductions very brief because most of them, I'm sure, are well known to the audience, and there is a beautiful speaker profile book that you can also refer to in the future. We're gonna start with Dr. Watts, professors at the RNA Therapeutics Institute at the UMass Chan Medical School in Worcester, Mass. I know he has contributed to the development of multiple oligonucleotides for ALS, and so we're very excited to have him as the first speaker in this important session in this afternoon. His talk is about kind of an overview and a comparison of different genetic therapies and their relevance to ALS. So thank you so much for accepting our invitation. It's fantastic to have you here.
Oh, thanks very much. It's really a pleasure to be here. This is a great meeting, as you said. I'm honored to be part of the lineup as well. So I hope you can see my slides and hear me okay. I'm, as Sabrina mentioned, I've really been tasked with giving an introduction to the session, not so much focusing on specific projects, but providing a bird's eye view of genetic therapies and their relevance to ALS. And gene-targeted therapies or genetic therapies are a very diverse group of interventions. And I wanted to begin by sort of plotting all of the different types of interventions along this axis of information flow within the cell. So we have our genes in the form of DNA, they're transcribed to RNA, they make protein, and at each stage of this process, there are ways we can intervene in a programmable way. Some of the approaches are conceptually quite simple. About 18 or 20 bases of RNA or DNA is sufficient to engage gene silencing or splice modulation. We'll come to each of these in more detail. Of course, many of us over the past couple of years received an mRNA drug in the form of as a COVID vaccine, a very different kind of molecule, 1,000 bases or more, a couple of thousand bases, but wrapped in a lipid nanoparticle. And then there are also important DNA-focused approaches, replacing a gene delivered by a virus or providing a virally expressed construct that affects cellular gene expression, or even editing gene expression. And so just to underline at the very beginning the diversity of these approaches, if we contrast one of the simplest approaches, an antisense oligo, that's 18 or 20 bases of chemically modified DNA or RNA delivered in saline. And on the other end, prime editing, one of the kind of next generation flavors of gene editing, includes about 7,000 bases of messenger RNA, 140 base prime editing guide, another 100 base nicking guide to increase the efficacy, and a formulation that consists of 4 or more lipids. And so the difference in complexity is like comparing a bicycle to a Lamborghini, and the differences in opportunities and mechanisms. But all of these diverse classes of therapies have something important in common. All of these are what we might call informational drugs. And there are two elements to that definition. First of all of these allow us to engage rationally and intuitively with biological information, with sequencing information, with information from studies in laboratories. And all of these have platform potential. And what do we mean by that? So for a traditional small molecule drug, the molecular features that determine their tissue distribution and stability come from the same set of atoms as the molecular features that determine target recognition and modulation. The word pharmacophore is used for the ensemble of molecular features that determine how a molecule recognizes a target, which polar groups here or hydrophobic groups here stick in which orientations and how far apart they are and how they bind to a particular protein target. That's called the pharmacophore, an old word that medicinal chemists use. Pharmacology bearer would be the origin of that word. So my colleague Anastasia Karova and I thought we really need a new word to describe this second set of features that has never needed a word before because it's always been tied up in the same set of atoms, in the same chemical structure. In an informational drug, that set of properties of molecular features that determines tissue distribution and stability can be separately optimized to a large extent. And so we named it the Dianophore, the distribution or delivery bearer would be the Greek origin there. And that's really driven by the backbone chemistry of the molecule, by the ligand, by the vehicle. It is not always perfectly separated. Antisense oligos of different sequences have different pharmacokinetics, but each time we improve the process, each time we learn something about the platform, we can apply it to other members of the same class of drugs. Antisense oligos probably have the smallest amount of separation between these two parameters. The Dianophore is affected by the sequence of the bases. But when we get to things like viral gene therapies or gene editing drugs in lipid vehicles, those do have really quite consistent pharmacokinetics, no matter which gene we're targeting, no matter what the sequence of bases is. And so what that means is that with each drug that's tested from a given class of the therapies we'll talk about this afternoon, we learn more about how to improve the next intervention. So as Merit mentioned, the field is seeing increasing rates of success. And part of that is learning ALS biology, but another part of that is learning how to improve the technology with each successive approval and even each successive failure. We can learn what went wrong and how to grow, how to redesign our drugs. Chemistry is important. Antisense oligos and siRNAs designed today have significantly higher potency and higher safety than the first generation of these molecules. And then many aspects of pharmacokinetics are shared across different members of a class of drugs, even though their sequences differ. And so we heard from Tim Miller and from Suma this morning about clinical trial designs and the importance of knowing onset rates of when we might see differences and how we design trials to avoid missing something that really is already happening. I meant to highlight when I came to this, this diverse mechanisms slide, it's an important day. It's a historic day because the very first CRISPR therapy was approved in the UK. There were examples previously of FDA-approved members of all of these mechanisms, all of these classes. The CRISPR drug that was approved today, of course, is not CRISPR editing in the brain. It's editing hematopoietic stem cells ex vivo and then reintroducing them into a patient for hematological diseases. But nonetheless, as of today, we can officially say that all of these 5 kinds of mechanisms have received regulatory approval. So there's significant momentum in the field, significant growth that compounds on additional growth. So we'll look at each of those 5 kind of mechanisms in more detail and in how they relate to ALS. But I want to, before we jump into each of those types, I just want to make one more comment. And that is that these genetic therapies, even though we think of them as being gene specific, they can be targeted at different levels of specificity and applied to different subpopulations of patients. So some of these we might imagine as mutation specific, applying only to patients with a very specific mutation in one gene. Others might be target specific, and so they might apply to all patients who have mutations in SOD1, for example, not just one type of mutation in SOD1, but nonetheless, only patients who have SOD1 mutations. And then a third level of specificity would be pathway specific or pathway focused interventions. So let's say, patients with TDP-43 pathology, a much broader pool of patients that may apply to sporadic cases or familial cases that are not yet fully understood. So let's look a little bit what we mean for each of those kinds of mutations. So to look first at mutation-specific interventions, many forms of ALS, especially familial ALS, can be attributed to very specific mutations. Some forms of sporadic ALS as well, of course. Increasingly, we understand the genetic cause. So let's take just one example out of many we could have chosen. Let's take TDP-43, TARDP G290A. That's a very different thing potentially than a patient with a mutation in a different gene or even a mutation in a different part of TDP-43. But these mutations may cause toxicity through multiple mechanisms. TDP-43 has both gain of function and loss of function toxicity, a pathology. And we often, when treating specific mutations that we haven't fully characterized, we need to make hypotheses. I think if I could get rid of the toxic gain of function approach from the C9orf72, polydipeptides, I could have a disease-modifying effect. Well, what else might be happening there? That's a disease-truant hypothesis that sometimes works out well, and sometimes we realize it's not the whole cause. And so treating mutations at a genetic level by reverting to the wild-type sequence, the healthy sequence, gives us the lowest dependence on a specific disease hypothesis. Maybe the highest potential then for a disease-modifying therapy. But of course, if we're going to treat each mutation at a time, that means we have a lot of interventions customized for specific mutations. Well, let's go one step bigger and look at target-specific interventions. And these would be examples where, for example, a mutant gene causes the disease through a toxic gain-of-function mechanism, in which case all patients with those mutations could be treated by silencing the gene. So tofersen is a great example of this kind of intervention. Or we could also imagine interventions where a mutated gene causes the disease by loss of function, including haploinsufficient mechanisms. And here you could imagine treating all the patients with insufficient expression being treated, for example, by upregulating a wild-type allele if they have one healthy allele. So that might apply to a family of mutations, but in a target-specific way. And then finally, pathway-focused interventions. Some cellular effects, of course, are common across many mutations in many genes, and are even seen when no genetic mutation is observed, as far as we know. So TDP-43 pathology plays a huge role for the vast majority of ALS patients. Neuroinflammation, again, may be significantly involved. And these are pathologies that can be addressed as a whole. And interventions that address elements of this pathology may be relevant and to a very broad population. And of course, some small molecule drugs, Amylyx's drug, taps into some of these broad pathways and is relevant to a broad group of patients. But I just wanted to highlight that our genetic therapies may also be relevant to these pathway-focused interventions, even though we often go quite quickly to the target and mutation levels. So all of these levels can be addressed with each of the different classes of genetic therapies that we will see, but some classes are particularly well-suited for each level of intervention. So let's now dig into those classes in a little bit more depth, and we'll take a couple of examples as time permits. So first of all, RNA silencing approaches, antisense oligos and siRNAs. These are molecularly the simplest approaches, mechanistically the simplest approaches. Really what we're looking at here is an anti-RNA agent that uses the same mechanism that our DNA strands use to stick together. A base pairs with T or U, G base pairs with C. So if we have a mutation in an mRNA of this sequence, the mRNA, of course, is thousands of bases long, but we can introduce a short sequence, 18 or 20 bases is sufficient. It acts as sort of a search query. It enters the cell, binds to complementary sequence. Not all sequences are as good as others, so we have to do pretty extensive screening to find sequences that are potent and safe, but it binds with base pairing. And antisense just means complementary, the opposite sense of the compared to the RNA itself, the messenger RNA. So this idea is so simple. It is not a new idea. It was first shown in 1978. Why are we only seeing tofersen approved this past year? What has taken the time for this to navigate its way to the clinic? Let's just take a little bit of a journey through some of the steps forward and how this points to potential next steps. So as I said, the first antisense experiments were published by Zemechnik and Stevenson in 1978. They synthesized a 13 nucleotide molecule of DNA, and they used it to show that they could inhibit viral replication in a model of Rous sarcoma virus. 2 papers back-to-back in PNAS. But this was an impressive study, partly because the first automated synthesis of oligonucleotides wasn't invented until 2 years later. So even making their molecule was a major project at this point. And the mechanism wasn't understood until another 6 years after that. The idea that DNA bases, when they're involved, recruit this enzyme called RNase H that recognizes hybrid duplexes, a duplex with one strand RNA and one strand DNA, and it cleaves the RNA strand of that duplex, allowing then the antisense oligo, the part with DNA, to go and bind another copy and cleave it. This is how tofersen works. And in the late 1980s through the 90s, and even since then, there has been steady progress. And in particular, chemical modifications have begun to become, the importance of chemical modifications has been really clear. And over the years, with good work from Tim Miller this morning and the group at Ionis and many others, it's become more and more clear how we modify antisense oligos so that they're reasonably well tolerated in the CNS. The first antisense oligo was approved in the late 1990s, in 1998, for an indication in the eye, a viral infection in the eye. But another surprise occurred also in 1998 with important implications for the field. And that is that a group of investigators led by Andy Fire and Craig Mello were carrying out a negative control. They delivered an antisense RNA into worms, which was their model organism of choice. But as a control, they put a complementary RNA onto the onto their antisense RNA. So it was already a duplex. It shouldn't have needed to bind a strand. And they found that this was more effective at producing gene silencing than either the sense or antisense strands alone. And that was so surprising and so important that it won them the Nobel Prize. And it really pointed to a biological mechanism that has become clinically useful. So an antisense oligo is delivered as a single strand. It finds its target alone and binds through base pairing. If it contains DNA bases, that recruits RNase H and silences gene expression. Later in the talk and in other talks today, you'll also hear about other mechanisms that can redirect splicing and affect other cellular processes as well if it doesn't contain DNA. But a small interfering RNA, because it is already base paired, it requires this cellular machinery. It's taken up by an Argonaute protein. Argonaute kicks off the passenger strand. And as you can see, and I've drawn this a bit suggestively, so that you can see that this is essentially an antisense oligo preloaded into a nuclease, into Argonaute. And that then guides the nuclease, guides Argonaute to the mRNA and leads to its cleavage. And so these are very parallel mechanisms, but they have very different biophysical properties and very different intracellular fates, intracellular levels of targeting. Antisense oligos work mainly in the nucleus. They can even silence co-transcriptionally. siRNAs work mainly in the cytoplasm. But they also have very different biophysical properties. And I want to just look quickly because this has implications for how we apply them to the clinic. So single-stranded oligonucleotides are really flexible. They have a persistence length of about 40 angstroms or 13 nucleotides. Persistence length simply is just sort of how far away you have to be before you stop noticing if somebody, bends where you started. And so a duplex has a persistence length about 10 times longer. It's 10 times longer rigid region. And so this changes which proteins it will bind inside cells in CSF. Single strands are also amphiphilic. They have hydrophobic bases on one side and polar phosphates on the other, whereas duplexes have all the non-polar bases on the inside of a stacked duplex, and all of the outside is polar, heavily hydrated phosphates. So it's very hydrophilic, whereas single strands are amphiphilic. And what that means is that single strands have quite high protein binding. So the bases and phosphates can arrange themselves in a line, can bind to proteins through multiple modes of interaction. Whereas duplexes have quite a bit lower protein binding. They mostly bind RNA binding proteins with electrostatic effects. And all of this translates into higher bioavailability for single strands than for duplexes. How does this play out in ALS therapies? Well, phosphorothioate modified antisense oligos can be delivered naked. Tofersen distributes broadly through the CNS. A number of other oligos have been in clinical trials or are still in clinical trials. Splice modulation oligos have very similar biophysics, very similar pharmacokinetics. And so they're able to be taken up freely by cells because they bind to proteins on cell surfaces some of which are trafficking proteins and internalize these. They escape the endosome by binding to endosomal surface proteins. siRNAs, in contrast, are dependent on conjugation or partly single-stranded designs, or in one approved case, a formulation approach for the liver. But in the brain, the approaches that are being tested are these conjugation or asymmetric approaches. So Alnylam has a design where they take a duplex siRNA that they conjugate to a C16 lipid partway along the sense strand of the siRNA. And that distributes pretty well in the CNS and shows robust silencing. In a partnership with Regeneron, they have multiple drugs, not for ALS, but for other neurological diseases, looking good. Atalanta Therapeutics takes a different approach and says, let's incorporate regions of single-strandedness with those flexible amphiphilic properties that allow them to bind to cell surfaces. And the larger size here also contributes to their retention in CSF. And one molecule that my group has found recently that's not yet published is these conjugates with antisense oligos with siRNAs that allows a slightly increased amount of single-stranded character and really robust uptake, robust PK, and allows us to silence or modulate genes through both mechanisms, gapmer antisense oligos or splice modulating oligos, coupled with robust gene silencing through the siRNA domain. So this can modulate two genes at once in a really very clean way. So we're encouraged about that. And another group actually in parallel just put a preprint out with a non-targeting oligo, just a random sequence of bases here that again shows an improvement in uptake in the CNS. And I think Bob Brown will touch on that in his talk later. There are a number of questions for gene silencing. Will this therapeutic index of antisense oligos and siRNAs continue to improve? Right now, the therapeutic index is maybe just about good enough, but a number of trials are still failing. Even late-stage trials are still failing because the therapeutic index is not comfortably wide. As chemistry improves, the therapeutic index does continue to improve, but we're not ready to stop yet. Another question is, will many targets require allele selective silencing? This dramatically limits the targetable sequence, and it's not clear how often it will be successful if we have to target at a mutation. Will efficient pathways to approval emerge for very small populations? And Tim Yu may touch on this later in his talk. He's been one of the leaders in these very nano-rare interventions. What targets for silencing will emerge in sporadic ALS? How will siRNAs begin to compete with antisense oligos as they gather an increasing amount of clinical data? These are key questions for the years ahead for silencing. The next kind of mechanisms I want to talk about are these gene augmentation and splice modulation approaches. And Tim Yu will really get into these much more in his talk. These are steric blocker antisense oligos. They don't contain DNA, so they don't recruit RNase H and silence expression, but instead, they can redirect splicing. So one great example of this field is in TDP43 pathology. Patients with TDP43 pathology have numerous missplicing events because TDP43 is not there regulating what it should be regulating. Genes like STAthmin2 and UNC13A have emerged as key targets that should have been corrected or correctly spliced by TDP43, but aren't. I'm going to skip fairly quickly through this. Let me just show one example of STAthmin2. This was shown by two groups, Don Cleveland's group and Kevin Hagen's group, in back-to-back papers in Nature Neuroscience, that without TDP43, a pseudoexon here is spliced onto STAthmin2 and causes, instead of full-length STAthmin2 being expressed, it causes this truncated form to be expressed. And we worked with Kevin's lab to show that an antisense oligo could be a surrogate for TDP43 and redirect splicing. Instead of splicing onto that pseudoexon, we could redirect it so that it forms full-length STAthmin2. So an antisense oligo, very similar biophysics to the gapmers, very different mechanism, and leads to rescue of STAthmin2 expression. I think I'll actually skip through the data here, but long story short, in the presence of these antisense oligos, neurons have the ability to repair their axons more effectively when TDP43 is silenced than they do with a non-targeting oligo control. A second level at which splice modulation becomes important is that deep intronic mutations can cause missplicing events. If they create a splice site, and that can lead to loss of functional gene expression. So patients with these disease-driving mutations might be treatable with the splice modulating antisense oligo. And Tim has pioneered this work in even a single patient, ASO designed for a single patient. That was Gleason, and it may apply to broader populations as well. So some key questions here. Will correcting the splicing of Stathman2 or UNC13A or any of these other TDP43 regulated RNAs be sufficient to bring disease-modifying benefit? Or will that just not be enough since TDP43 is normally regulating so many genes? And then can we find pathways to facilitate diagnosis and treatment of patients with ultra-rare, perhaps deep intronic loss of function mutations and find ways for them to get patient-specific ASOs? And then finally, in the last couple of minutes, I just want to look at some challenges and opportunities in DNA-focused approaches, viral gene replacement therapy, other viral therapies, and then gene editing therapies. So for viral gene therapies, we're using, of course, a repurposed, usually an AAV virus to express a gene. Typically, this is, when it's gene replacement, this is replacing a gene to treat loss of function pathology. Another approach with an AAV can be to express a gene silencing agent, something like an siRNA. Bob Brown is going to talk about a microRNA to silence SOD1 in his talk a little later. In both cases, that can be a one and done approach, so a single intervention with a durable effect. But there are some ongoing questions. Will this transduction and gene expression be sufficiently homogeneous across brain regions and cell types? Huge progress in these areas as new vectors come. We'll hear more, I think, about this from Jerry Mendel in his talk. And then how will expression last over potentially many years? This is an ongoing question that can only be answered, I think, by increased clinical experience. And then two types of gene editing, of next generation gene editing, that are increasingly important. So base editing is highly potent and highly specific for correcting a very small subset of mutations. So transition mutations, turning A to G or T to C, is pretty well established to be potent and specific by work from David Liu's lab. Earlier work from his lab that's not quite as mature in terms of specificity can also do C to T and G to A. These are all transition mutations, and there are many mutations that don't fit into these classes. Patients with those mutations, that might be very appropriate on a patient-specific intervention basis. But there may also be ways to apply this technology to targets and pathways and pathologies. So this is something for us as a field to reflect. As base editing platform technology approaches the clinic, how can we, as an ALS community, make the best use of it for the broadest number of patients? So for the one mutation at a time approach, how do we make sure that patients with ultra-rare mutations can benefit? Base editing can be delivered through an AAV. There are compact base editors that work in a single vector. Is that safe for the long term to be expressing a Cas9 editor, or does that lead to off-target editing? Or can non-viral delivery, transient delivery, achieve robust editing in deep brain? Those are both areas that are significant works in progress by multiple groups. And then finally, there's a very exciting and versatile gene editing technology called prime editing, in which Cas9 is paired with a reverse transcriptase or other polymerase. And this could really apply to correcting all small mutations. It may be able to correct a family of proximal mutations, a hotspot with multiple mutations, so that patients with ultra-rare mutations may not have to wait for a single patient customized approach. And they can even provide pretty robust repeat contraction. So for C9orf72, for example, a pair of prime editors on either side of the repeat can really drive robust contraction of that repeat. But there are a number of key questions. Prime editors are bigger because of the fusion with the polymerase, and so they don't fit into a single AAV. Paired AAVs, dual AAVs may be okay, but also for this approach, it may be dependent on transient non-viral lipid-based delivery approaches or virus-like particle delivery approaches to achieve robust editing in deep brain. And we're just not there yet in terms of the technology. So will this editing efficiency in vivo? There are other elements besides delivery for prime editing that are still works in progress. It's not to the level of being therapeutically relevant yet. So I'll leave you there. I hope that this summary has been helpful to sort of lay the groundwork for the talks ahead, where the speakers will be getting into their specific work in all of these areas. And hope you'll agree, it's just a really exciting time to be in this space. Thanks so much for your attention.
Thank you so much. I mean, this was honestly the best overview I've ever seen of this emerging field. I feel like I did my PhD in the Stone Age, clearly, when just when the sRNAs were just being developed, and to see the advances that have happened and the way you explain them and the potential, really, what's interesting is obviously I'm in awe at the complexity of the biology, the technological developments, but most importantly, the potential for interventions for many different types of diseases, as you clearly pointed out, both in terms of, ultra rare mutations, but also going into sporadic disease by the ability of modulating pathways. So this is incredibly exciting. I know that we are a little bit behind time, but for good reasons, because this was an important overview, because it really lays the groundwork for the next topics and talks. So I think we can move on to the next speaker, but if there are questions, it would be great to keep your questions for the panel. Again, at 3:30, we're going to have a great panel with the speakers. And now